Our next guest speaker is Dr. Fajarudin Ahmad from Research Center for Biology, National Research and Innovation Agency, or BRIN, in Indonesia. He had a bachelor and master degree in plant genetics and PhD in experimental plant science from Wageningen University Research. He has been working a lot with the polyploid plants such as potato and, of course, banana. Today, he will talk about the gene mapping towards banana genetic improvement, breeding for Fusarium wilt-resistant banana. Welcome, Dr. Fajarudin.
Thank you, moderator. Share my screen. Okay, thank you. First of all, I would like to thank to the CAB and KUIS International Seminar Series Committee for the invitation for this occasion. And I would like to greet all participants and speakers. And this morning, I'm going to present a topic about gene mapping towards banana genetic improvement. So in case of for breeding for Fusarium wilt resistant banana. So before I want to go to the main topic of my presentation, I want to refresh a bit the importance of banana. So banana is one of the most important and popular fruit, not only for people in tropical countries, but also in subtropical countries in Europe and in America. We can enjoy the fruit as a fruit, we can bake it, or we can make it cheap as a snack. And recently,
Sorry.
Yes?
Sorry to interrupt, I haven't seen the your slide yet.
Oh, really? Wait a minute. Okay. Okay, can you see my screen?
Yes. All right, yes.
Okay, sorry for the trouble. Yes, the banana is one of the important fruit that the production is recently exceed than 120 million ton in a year. Can imagine the economic value of this fruit. Approximately, there are more than 700 cultivars around the world with various size and flavor with various genome from AA and hybrids AB or from diploid, triploid, and tetraploid. However, only few cultivars are extensively cultivated. Mostly is from Cavendish group and followed by plantain and Gros Michel. And recent days, banana production is facing some challenges, mainly by biotic stress, pests and disease, and also abiotic stress because of climate change, such as drought. The most devastating disease is Fusarium wilt. The infection is initiated with the penetration of the fungus from the root and block the vascular system. So that's why it's resulting wilting and discoloration appeared in the pseudostem and the rhizome. And this disease, as mentioned by the previous speaker, that it's caused by Fusarium oxysporum cubense, and recently also described it as Fusarium odoratissimum. And this disease has become worse because the fungus can produce the chlamydia spore that can stand in the soil more than 30 years. And based on the host and disease compatibility, there are four races. The race 1 is compatible to the Gros Michel group, race 2 is compatible to the blue group, banana group, and race 3 is no longer described as a banana disease because it's only infected ornamental plants, heliconia. And the last one is race 4, especially tropical race 4, is compatible to majority banana cultivar. The disease have changed the major banana productions after the Gros Michel banana industry was collapsed in 1950s. In the beginning of the banana industry, the gross missile is the most produced by banana companies in tropical America in 19th century. But since the malady occurred and not possible to replant the banana in the same soil, then farmers changed the banana to Cavendish, which resistant to the disease. The latter that the disease is identified as Fusarium oxysporum race 1. And since that time, the production of banana was growing until a new races of the disease discovered and attacked the Cavendish. The recent race of the Fusarium is tropical race 4. And recently, tropical race 4 reported found in Latin America. Hence, we need to increase our awareness to the possibility of repeating history of the cruise missile in the past. Resistant plan is needed to keep the sustainability of the banana productions. This means developing the Fusarium wilt resistant banana is needed in the near future. Since the beginning of banana industries in 19th century and the present of the disease, especially Fusarium wilt, the initiation of banana improvement has begun. The initial attempt is classical breeding and followed by some approaches such as mutation, somaclonal selection, transgenic, and recently the development of gene editing using CRISPR-Cas is promising for genetic improvement for banana. Classical breeding and mutation is not required gene identification preceding the activities. In contrast, transgenic and gene editing require gene identification preceding the work. I'm thinking that in the future of banana genetic improvement will be more efficient, targeted, and using recent technology such as gene editing or CRISPR-Cas. These approaches need information about the genes that associated to the desired traits. And mapping is an approach to identify the gene or region that associated to the desired trait. An advantage of mapping is this method can provide straightforward information of markers that associated to the desired traits on the chromosomes. Afterwards, marker also can be developed as marker-assisted selections that's specific to the disease or any traits that we want to identify. And this is the general flow of the gene identifications. So there are 3 stages: mapping, verification or validation, and isolations the desired genes. So the first stage is mapping. So this is the stage that I would like to present more detail in this morning. So the mapping is begin with the creating a population. We can cross or selfing to create the segregation population or to accumulate the desired allele. From this stage, followed by phenotyping and genotyping to construct the linkage map. Phenotyping can be done in the greenhouse bioassay or in the field. And then the genotypings, there are many markers available in this moment. There are AFLP, DART sequencing, SNPs, microsatellite, and many other markers. And combination of phenotyping and genotyping data, we can analyze this for fine mapping the markers that associated to the desired traits. And afterwards, the second stage, we go to the verification and validation. The QTL or markers that we obtain from the fine mapping need to be verified or validated to make sure that the markers are really associated to the trait. Some method has been developed or some can be used to approach to verify the markers such as overexpression, suppression, knockout, knockdown, So I think overexpression is the most simplest way we can we just isolate the RNA and then we check the expression of the susceptible, comparing the susceptible and resistant plants, and so on. Once we verified or validate the markers, and then we can isolate the candidate genes. Once the candidate gene isolate, we need to validate the gene if it's true associated to the desired trait. At this moment, CRISPR is extensively used by researchers because it's relatively easy and targeted, and we can use it cheaper and cheaper day by day now. So how to validate? We insert the gene into targeted accession or genotypes. If we want to assess the resistance, we can insert the gene into susceptible plant if the plant's getting resistant. So it's confirmed that the gene is responsible to the resistance. And once the gene is validated, we can use the gene for any genetic improvement with this kind of biotechnology for transgenic or cisgenic for other bananas, or also design and consider for breeding strategy. And from this gene, we also can develop the marker assist selections to efficient the breeding program. And actually, the marker assist selection not only can be built from the genes, but also from marker that we obtain from the fine mapping to develop the molecular assist selections. In the next slides, I would like to present our works on mapping of Fusarium wilt of banana resistance from an accession of wild Musa acuminata. So this is the banana that we use for the mapping, Musa acuminata subspecies malaxiensis, accession LIPI 010. So this banana is considered as a heterozygous resistant to Fusarium wilt banana. So we use this. So we self this banana to create the segregation populations, because this one is heterozygous. So the population, the segregated population, is equal to F2 populations. And why we choose this banana? Because the one of relative is accession malaxiensis double haploid pahang has been whole genome sequences, and the sequence are deposited in the gene bank, and then we can use the sequences for the reference. And this works already published in Theoretical Applied Genetics in 2020. You may check it. So this is our step for the mapping. We started with creating the segregation population by selfing pollinations. We select 255 genotypes for greenhouse bioassay and genotyping. So the genotyping is based on dot sequencing, and from 32,000 markers retrieved from these markers, we filtered to 2,800 SNPs marker, and from this selected marker and then the phenotyping data from the bioassay we use for develop the linkage map. In this works, we map two races of Fusarium wilt, race 1 and tropical race 4, the two most important races for the banana cultivation. And in this thesis evaluations, we use the leaf symptom, the wilting symptom, and the discoloration of the rhizome. So we divide it into score 1 to 6. 1 is for the symptomless plants, and 6 is the worst symptom plant. And this is the result. This is the level of severity for race 1. The distribution of rhizome severity score is like a slope, and then the distribution for leaf severity symptom is like is bimodal-like distribution. So this result indicated that the RIS1 is resistant single dominant. And in contrast, the pattern of rhizome and leaf score are normal-like distributions that indicate there is a gene interaction for the tropical RIS4 resistance. And to see the marker associated to the resistance, and we would like to know which chromosome pair is resistant. We did the interval mapping, and then we set the threshold of the LOD, and we found that the markers that above the LOD is found on chromosome 10 that indicate the resistance of the RIS1 and tropical RIS4 is on chromosome 10. And this is the more detail of the interval mapping. So I combine all the leaf scores, rhizome score based on RIS1 and tropical RIS4. Here we notice that the RIS1, the markers that associated with the RIS1 look obviously significant on chromosome 10, but less significant not for the tropical RIS4. And because of the rhizome score is much higher than the leaf score, so afterward we consider the rhizome score for the mapping. And according to the average score of the rhizome and leaves, there is one, as I mentioned before, that this is single dominant. This is obvious from the pattern of the rhizome score distribution. When the TR4 resistance is additive, neither dominant or recessive. So when once the RIS1 occur, the RIS1 resistant occur, the resistance to the tropical RIS4 is increased. But when the resistance is not occur in the allele, and then the susceptibility of the plants to tropical RIS4 is increased. As I mentioned in previously, that the fine mapping only done for the RIS1. So this is the fine mapping for the RIS1 resistance. Because of the significant score, the LOD is obviously above the threshold. Then we could perform the fine mapping. And eventually, we found 4 markers that associated to the resistance at the tip of chromosome 10 out of 106 marker above the LOD. And in this region, we identified 19 genes or predicted genes out of 164 genes that associated to the resistance based on double haploid Baham sequences. So the further works, I need to continue this work, which is not done yet, is the verification and validation the marker associated to the resistance to develop the marker-assisted selections, and also verification and validation the candidate genes in the region of chromosome 10. So this is the 4 sequences on tip of chromosome 10 that highly associated to the resistance. I'm thinking to develop this marker for molecular assist selections. So the sequence is there, so I can design a primer. So I'm going to apply it to the resistant and then susceptible plants, and to see the response of this marker to the resistant and susceptible plant. And here is the part of chromosome 10 sequences. So we did resequencing the resistant plants and susceptible plants, the homozygous on that resistant region. And from our first impressions that there is a difference between the sequences of the resistant plant and susceptible plant. To make it simple, I make a diagram here. So I make a block, a green block. So this one is the block for the resistance. So the green is a gene. So I assume the green block is genes, and the susceptible one, they miss 1 or 2 genes here. So I'm thinking to verify this the occurrence of the gene using knockout or knockdown or overexpression as well, to verify that the genes is truly associated to the resistance. Once the genes is verified, I would like we can develop the molecular acid selection from the genes, and we can isolate the gene, and we will continue for the transformation or gene editing. And mapping also enable study to the structural arrangement of the chromosome. So this is the our research, the result of our mapping. From this work, we confirm there is a structural arrangement between chromosome 1 and chromosome 4. So if you notice here, the blue is part of chromosome 4 and the red is part of chromosome 1. So when we map the marker to the chromosomes of banana in our populations, some part of chromosome 4, they are part of the chromosome. Some part of chromosome 1, also part the chromosome 4. So it's confirmed that there is a translocation between chromosome 1 and chromosome 4 in subspecies Malacensis accession DP010 that we use for the mapping. Mapping is quite promising. Provide a straightforward result to the targeted region or marker or gene that we would like to study. But mapping is still relatively costly for a researcher in developing countries such as in Indonesia. Besides, mapping require fertile banana, and most banana cultivar is sterile. However, banana wild relative, Musa acuminata and Musa balbisiana is available and grow and exist in the center of diversity in Southeast Asia, such as in Indonesia, Thailand, and surrounding. We still find this species in forest border and the countryside, the villages. It's not that difficult. And these species are fertile, so we can utilize this banana for mapping. So identifying gene from fertile but relative is possible, as I presented in these works. And I'm also thinking about the gene pyramiding program in the future. So we can make deploy it from wild improve that contain more than one desired genes. So I think in the near future, we need to do much more extensive of exploration and evaluation of banana wild relative. This is the example of Musa acuminata, wild Musa acuminata in Indonesia that is described by Nasution in 1991. At least there are 15 varieties, and we also found Musa balbisiana, Klutuk bulung in Indonesia. But the diversity of Musa balbisiana is not as high as Musa acuminata. And these are a few potential wild Musa acuminata that can be considered for a mapping study in the future. Again, subspecies Malacensis that already known as resistant to Fusarium wilt, and in our early screening that we did a few weeks ago, we found there is an accession of phenotypes that resistant to banana plant cytop disease, and we also found a few cases that the fruit is developed without any seeds development inside. So I think this subspecies is promising for parthenocarpy study. And the other one is subspecies sumatrana. I think we can consider this Musa acuminata for agronomical traits because the bunch appearance is look nice. And in a bunch in natural habitat, I found a bunch with up to 25 annuals. So it's really nice for banana breeding program in the future. Okay, so I think I'm in the end of my presentation, but before I finish my talk, I would like to thanks to people that contribute to this work, my supervisor, and here also present Hans and Pufu, also Professor Fatima and Hank Sultan and Dr. Liu from Libya, and also sponsor and institutions that contributing to this work. And I would like to thanks for your attention. Thank you.
Thank you, Dr. Fajarudin, for sharing with us. Anybody have the questions? Well, Dr. Hugo again? Okay. Go ahead, Dr. Hugo.
Yes, please.
Hi, Fajar.
Yes, hi.
When I look at your disease score graphs, the Fusarium, the tropical race 4 is a normal distribution, let's say. The race 1 is something I have not seen before. Very strange, but none of them seem to be a qualitative trait. So there seems to be multiple genes involved to get this kind of distribution.
Do you mean for the both assistant race 1 and triple race 4?
For both of them, it's if you have a qualitative distribution, you see clearly resistance and susceptibility, and there are two groups. Here you see a whole range of different levels of resistance and susceptibility. So that indicates there is a quantitative trait. So there are multiple genes involved. How many, I don't know. So you may have found one region. Where's the rest?
There is one.
Both race 1 and race 4 seems to be more a classical pattern.
Yes, it's more like a distribution, but interestingly, it's only on chromosome 10. But yes, we are a bit still thinking about the tropical race 4 because the LODs is not that as high as the race 1. So we're thinking about there is still something need to be studied more on the tropical race 4 in this accession LIPI 0 LIPI 10 is this one. So in that moment when I finished this work, I also discussed with one researcher in Ur, Heng. So he is also contribute to this research. So probably in this accession is cannot solve very well to solve the resistance for the tropical race 4. So and the possibility of the interactions of race 1 resistant to and tropical race 4 may influence our analysis, but we still have no idea about this. Ideally, we did at least 3 biological replications in this work. I only did one biological replication because of times and budget considerations. So we get to make sure that the population and is respond to the disease is because of the disease, not because of the environment situation. I did these experiments in quite long periods. in 1.5 years. So I passed the spring and summer, winter, and so on. And I think, yeah, it might influence a sort of the experiment, but as we try to make the experiment as best as possible in the similar situation. Yeah, I'm thinking of to do the further study for this cocoa, not only for lipid 10s, but we also have another accession in our institutes, and that's look promising as well for the Fusarium wilt mapping.
I also noticed that in your mapping results, your Manhattan plots, let's say, there are quite some significant results in the unmapped scaffolds.
Yeah.
Have you tried to take some of those results and try to map them on the unplaced? Does mean they cannot be put into the reference sequence at the exact location, but maybe we can map them to a particular chromosome region anyway?
Yes, recently, yes. When I finished this manuscript, there is the available sequence is the version 2, I think. Yes, the version 2. And after that, after I finish this, I just try to blast the sequence from the unanchored markers, and I found that one or two, I don't remember by heart, but one or two they can be matched in chromosome 10. Match chromosome 10. There are about more than 100 marker, I think, in the unanchored at the time. So I didn't check one by one. I just randomly put, okay, it's work. So it's confirmed in chromosome 10.
All right. I have a little questions. When you, I saw that when you scoring, you have a scoring result of the rhizome from the leaf and somewhere else. But when you do the inoculation, did you do it at each point, or you just did from the rhizome and observe the disease symptom?
Sorry if I understood your question is what do you mean how I inoculate?
Yeah, you inoculate at what part of the plant when you do the inoculation at what one
so
place?
I inoculate the plant this is a 2 months old plant. The size is about 60 to 80 centimeter in height. So the diameter of the stem is about 3 to 4 centimeters. So I inoculate the plant. I'm using 1 million spore in a milli, and then I pour about 100 milliliter spore. I pour on the pot. But
on the pot, but not on the top of the plant, just on the soil, right?
No, because the Fusarium is in the infection is started from the roots. So
okay, that's why you said that the best symptom that you can say it's in the roots, right? It's from the roots.
Yes, yes it is.
But well, when you score the roots, I assume that you have to terminate the plant.
Yes,
right?
I have to.
So you can score the root just one time, right?
Yes.
How can you know that so you probably, if you score it one time, you might get just the major yin, and but if you score somewhere else, you can make a sequential score. You might get minor yin, right?
Yes, for each genotypes, I prepare about 5 plants as a replicate. So I score 5 of them. So in one genotype, the score is not exactly the same. Sometimes one plant is 1, I scored as 1, the other one is 2. So and then I average, get the average score of 5 points.
Thank you.